Hello, everyone. My name is Erola Fenollosa, and I'm a Marie Curie Fellow at the University of Oxford. It's a real pleasure and an honor to be here with you today as a keynote speaker. I want to start thanking the organizers for the invitation and all of you for being here. Due to an overlap with other academic activities, I can't be there in person, but I'm very open to continue conversations virtually if you would like more details. What I will share here today builds on our shared foundation on demography, but through a slightly different lens, how images and artificial intelligence can help us capture demographic processes in invasive plants, and how these tools might open new possibilities for demography. In this talk, I will be answering this question. Is it possible to extract key functional traits and demographic parameters relevant to invasive species performance and spread from images? Here you can see a brief outline of the talk, which after presenting the threat that biological invasions represent, I will discuss this imaging revolution and how it can contribute to measure survival, growth, and reproduction. Some of the projects I will present are ongoing projects that contain this symbol here. Feel free to reach me if you'd like to collaborate or are you interested in knowing more about those. Biological invasions constitute one of the main causes of biodiversity loss, and their numbers keeps increasing, which are especially high in plants. On the right, you can see examples of two major plant invaders threatening the Mediterranean biodiversity hotspot: the prickly pear belonging to the Opuntia genus and the cave pig belonging to the Capparoota genus. Demography for invasive species is crucial. We need to predict their presence, understand the areas where they can grow in order to prioritize management. However, demographic methods are data hungry and labor intensive. Current approaches include using quadrats to quantify abundance or tagging individuals and traveling to the study site. Here is where AI and imaging appears. Even from space, we are nowadays able to observe the Earth at 1.8 meters resolution in the world view. And even with this little drone, the DJI Mini 4, that you can also see here in my hands, anyone can collect centimeter level imagery with a 48 megapixel camera for under €1,000. The drop in cost and the leap in resolution are completely changing the scale and detail of the data we can access. And it's not just the hardware. On the software side, algorithms are now everywhere. Open source, well documented, and increasingly easy to use. Many of them don't even require specialized GPUs to run, meaning we can analyze imagery on standard laptops or in the cloud. Together, this combination of high resolution, affordable imaging, and accessible AI has democratized what used to be exclusive. It means we all can generate and analyze the kinds of data sets that we were unimaginable just a few years ago. Just as a reminder that when we talk about artificial intelligence, it's something very broad, it's something very open, of building machines that can perform tasks that we classically associate with intelligence. Within it, machine learning refers to algorithms that improve automatically with experience. And deep learning is a subset of machine learning that learns its own features directly from the data, making it particularly powerful for recognizing patterns in images. Images are structured data. Each image has a set of attributes that carry biological information we can use for demography. Even only within the visible range, what our eyes can see as green or red can be decomposed into numerical values in different color spaces. In the familiar RGB space, red, green, blue, we measure red, green, and blue channels. But we can also transform images into color spaces like HSV, where hue captures the dominant color, saturation reflects intensity, and value relates to brightness. Depending on what we're interested to see, we would choose those different color spaces. Spatial and spectral resolution will define also which elements are we able to see due to their size or to the wavelengths that they are reflecting to. With all those properties, we will be able to acquire survival, growth, and reproduction if we know how to read it. And this is what I'm going to explain now. Let's start with survival. By detecting individuals in images taken at different time steps, we can actually track their presence or absence. If an individual is detected at time 1 but not at time 2, this absence can be understood as mortality. In this way, object detection across repeated images provides a measure of survival. In a review we published this year, we reviewed all the literature using AI for invasion biology and found out that detection, and especially in terrestrial plants, is one of the main applications. In this example here is a very good case of usage of imaging for acquiring survival data. So survival from images is based on this temporally matching the individuals from two pictures. Um, in here, they use drones and calculate the spatial overlap between individuals from 2015 to 2019. Also, the timing of the census is very important since detection can be impossible at certain times, depending on the species phenology, as this case with Acacias ligna, which can be better detected during the flowering period. The different channels of the HSP color space can help distinguish species and individuals. If we couple imaging with deep learning, we can further cover more area. For example, in this recent work, we collected images of an invasive species in Spain, this one here, and used true-prong detections to train a deep learning model and further detect individuals around the study area. We covered this little area here, but through deep learning, we were able to cover all these areas around. Accuracy resulted very strong, even when using Google Earth imagery that doesn't have that much resolution. It has 19 centimeters per pixel by detecting mainly the biggest individuals. So the 80% of individuals were quite big and were actually detected with an 80% of accuracy. Here's another example of drone usage coupled to deep learning to detect and mask individuals, in this case using semantic annotation, with examples of the input and the predictions that the model did. So you can see it quite uh, accurate, represents and selects and masks the individual in these different images by using only this little drone here. If we move on to growth, images don't just tell us whether an individual is there, but also let us measure size changes. The key method here is segmentation, where algorithms outline the exact shape of each individual in an image. By comparing those segmented areas across time steps, we can quantify how much individual has expanded or shrunk. In practice, this means overlaying the segmented outline from time 1 onto the image from time 2 and calculate the difference. Of course, using images to measure growth isn't entirely new. Classically, ecologists have used them to obtain traits like leaf area, thickness, or shape, using ImageJ and programming macros to speed up the process. What is new is that with segmentation and overlap analysis, we can now scale these measurements up, from single organs or individuals to entire populations. These methods, as we were saying, are not restricted to close-up images. We can transfer the same principles to georeferenced aerial imagery collected by drones or satellites. With this approach, we can measure population growth or even track individual size changes, depending on the resolution of the imagery. As we can see in this example, here is the satellite image and a drone image that with very different resolution. In addition to the resolution, a critical piece here is accurate georeferencing. If images are not perfectly aligned across time, then a pixel-by-pixel temporal comparison wouldn't be reliable. High-quality ground control points and stable flight protocols are therefore essential for turning aerial images into demographic datasets. Here you can see the beautiful pipeline proposed by Alice Rosen from the lab, who fit an IPM contrasting eucalypt crowns over time to fit growth functions, being able to include around 40,000 individuals, which is quite a lot. Note that these methods are based on things that are sessile, things that are on the same spot. So it doesn't apply to other taxa that move, but some other taxa may have some elements that can be tracked. such as nests. At the end, we can discuss a bit more about this, how this can be transported to other taxa. Segmentation doesn't need to be manual. The SAM model, the Segment Anything model, which is open access, can be implemented in several softwares to segment automatically images after encoding them. This example here, this picture, shows segmentation of the invader Opuntia stricta in Greece. The algorithm extracts the outline of each plant, allowing us to estimate the area. With 3D information or a structure from motion photogrammetry, we can also estimate volume, which brings us closer to measured biomass. In this regard, I would like to recommend you the talk T12, Expanding this Time of Measurements. Beyond area, multispectral imaging has proved to be able to provide biomass estimates. For example, in this recent work, the authors calibrated the biomass model based on vegetation indexes for this invader. We aimed to include the author's work into our own demographic framework, and we started this collaborative work. We are trying to fit demographic models based on the pixel by pixel analysis and based in biomass as well. By comparing the image by 2022 with another one 2 years later, we are checking how much green biomass is still present. And we are defining recruitment, survival, mortality, and growth, depending on how the pixels turn out on the time too. This work is in preparation and further data processing is still required, but you can see here that it's quite interesting how recruitment is quite low, whereas we can see a lot of mortality in different areas. Not only survival, but also stress levels can be inferred from color, as we show in this recent article I published this year, where we feed regression models with machine learning algorithms to predict stress level from leaf level pictures in invasive plants. Finally, about reproduction, how we can estimate reproduction for images. We might have two different key approaches. So one would be classification. For example, is this an individual in a reproductive stage or is it not? Or also counting the reproductive effort itself. This is particularly powerful because counting reproductive units manually over large areas is extremely labor intensive, especially with invasive plants. By detecting flowers, buds, or fruits, algorithms can assign each individual to a developmental stage. For example, juvenile or adult or reproductive adult, as it was done in this case with this wheat on agricultural fields. These type of algorithms have been quite useful to detect flowering times as well, and those kind of algorithms can be also used for detecting fruits. In this regard, I would like to recommend talk 26 of the conference, which presents multi-species grassland demography that will be paired with imaging. In order to quantify reproductive effort from images, there are lots of solutions that come from smart agriculture, and there are a lot of applications such as Fruit Scout or Fruit AI that estimate or count directly the number of fruits. In a recent article, Meng Swan et al. Compared alternative approaches to count fruits, such as T-Rex. T-Rex is a foundational model where we would define a few elements of what we want to count, and it will automatically count the rest, as you can see in this example with these berries. Surprisingly, very good results were also obtained when asking directly ChatGPT. Images can also provide size estimates of fruits. In the same way we measure individual plant area or volume. This allows us to combine counts and sizes, giving a measure of reproductive effort, as we can see here with this apple example. So at this point, I would like to put everything together, but highlighting how we can use public data sets. So we know that the world is being imaged every day. So what if we could transform those images into demographic data? When I was last year doing fieldwork, I realized that one of the populations I was visiting could be seen by Google Earth imagery. I started exploring if that the patch of one invasive species I saw in 2024, you'd see here, could be seen and tracked back in time from those images. And surprisingly, yes. If you can keep your eye on this patch here, you can go back in time and check that this was there even like 18 years ago. With this in mind, we developed a pipeline accompanied by a set of video tutorials on how to perform retrospective image analysis for long-term demography using Google Earth imagery, which can be coupled to deep learning to automatize the process. Here you have a QR code to access the preprint. We tested this method of segmenting individuals, individual matching back in time, converting this into recruitment data, and then performing spatial-temporal explicit demographic analysis, not only with Opuntia that I presented earlier, the pre-clip here, but also with Carpurotus, the feed cap. For example, for the Opuntia population, for the prickly pear, we were able to obtain an 8-year series of recruitment data. By tracking individuals back in time, we were able to determine their age, or in some cases, minimum age, and characterize population structure, as you can see here, quantify high and low reproductive years in terms of recruitment, and explore the relationship between area and size, as you can see here, and identify areas and map areas with high and low recruitment, where we can identify where the youngest individuals are. By repeating this method in multiple ecosystems, we could have an estimation or the first approach into explore environmentally driven demographic plasticity. With the Caprodus species, we also use the Google Earth method to go back in time and track the species area. For this species that has clonal growth, this is quite important. And we saw these expansion patterns from 2007, for example, for this individual that keeps expanding with time. So we could fit models to measure or estimate mean annual growth of this species and actually also quantify the variability, the massive variability in growth that these shapes can show. Another source of imagery data beyond Google Earth is Google Street View. I have recently been awarded with a small grant from the British Ecological Society to develop this project entitled Street View Sentinel that will offer a tool to automatically detect invasive species from Google Street View images, starting with Opuntia, the prickly pear, in Sicily. The first deep learning algorithms trained are giving very promising results, although I can tell I'm surprised since the amount of Opuntia in Sicily Island is truly impressive. From the labeled image since the present day, we found a total of 2,306 individuals in only 1,120 images, with images showing up to more than 100 individuals. Since Google Street View has also temporal coverage, this dataset will allow us to test temporal dynamics in the island. However, there's plenty of data processing to transform the data to demographic insights in this case. What leads me to the next slide. While the potential of images for demography is enormous, there are important challenges to consider. First, there is data uncertainty and bias, as images are rarely perfect. Detection of individuals can strongly depend on image resolution and the surrounding vegetation, with occlusion creating gaps and biases in the dataset. Similarly, imperfect georeferencing can introduce substantial errors when estimating growth or survival. Using deep learning for automatic detection also has limitations. Training models often require extensive dataset labeling, which can demand expert input to delineate many individual instances. Looking ahead, there are several promising avenues for overcoming those challenges. Integrating image-based data with classing ground measurements can help reduce bias, for instance. Beyond trying to quantify demographic parameters for terrestrial plants, other elements that are fixed can be used to fit demographic models, or even quantifying population size for images is possible in moving animals. Transfer learning is a method that allows us to train models to detect fruits, flowers, or individuals, even with limited labeled data based on pre-trained models. Publicly available imagery opens the door to real-time monitoring of invasive species, a game changer for early detection and management. Finally, images are uniquely suited for studying especially explicit demography. They allow us to test density-dependent responses and microenvironmental variation, which are key for fine-scale demographic predictions and understanding population processes in complex landscapes. To finalize, by pairing images with AI, we can turn pixels into population processes at higher scales, speed, and high reproducibility. I would like to thank my collaborators and lab members, who I miss a lot, for their massive help and support, even when I'm far away collecting images, and all of the co-authors involved in the data presented here, as well as the funders beyond the different presented projects. Pixels, plans, and predictions. Let's talk about turning images into population insights.